Welcome to this introduction to general bacteriology. We'll explore the fascinating world of microorganisms, focusing primarily on bacteria. These tiny life forms come in many shapes and colors, and understanding their diversity is key to grasping how they impact health and disease. This sets the stage for our journey into the microscopic world that influences so much of our environment and medicine. Let's look at how medical microbiology branches into specialized fields. Each area focuses on different microorganisms or systems, like bacteria, viruses, fungi, or the immune response. Understanding these interconnected fields helps us see how complex and integrated the study of microbes really is, and why collaboration across these areas is essential for medical advances. Now, let's talk about some pioneering discoveries that laid the foundation for microbiology. It all started with Antony van Leeuwenhoek, who built the first microscope and was the first to observe microorganisms. Then, Louis Pasteur's germ theory revolutionized our understanding of disease, introducing sterilization and vaccines. These breakthroughs transformed medicine and opened the door for modern microbiology. Building on those discoveries, Lister and Koch made huge contributions. Lister introduced antiseptic surgery, which dramatically reduced infections. Koch established bacteriology as a scientific field, developing staining techniques and pure culture methods. He also identified specific bacteria causing diseases like anthrax and tuberculosis, and formulated Koch's postulates to prove causation. These steps were crucial in linking microbes to disease. Koch's postulates offer a logical sequence to prove that a specific microorganism causes a disease. First, you find the organism in diseased tissue. Then, you isolate it and grow it in pure culture. Next, you introduce it to a healthy subject to reproduce the disease. Finally, you re-isolate the same organism from that subject. This methodical approach is fundamental for understanding infectious diseases. To visualize Koch's postulates, imagine a cycle. The microorganism is consistently found in diseased animals, isolated in pure culture, then introduced into healthy ones to cause the disease, and finally recovered again. This cycle confirms the organism's role in causing illness, providing a clear experimental framework for disease causation. Classification divides living organisms into prokaryotes and eukaryotes based on cell structure. Prokaryotes are simpler, lacking a nucleus, while eukaryotes have complex cells with defined nuclei and organelles. Understanding these differences helps us grasp how bacteria function compared to more complex organisms, which is essential for studying their biology and behavior. Comparing eukaryotic and prokaryotic cells reveal many differences. Eukaryotes have larger cells with nuclei and organelles, while prokaryotes are smaller and simpler. Prokaryotes generally reproduce faster and have unique DNA structures like plasmids. These distinctions affect how organisms grow, replicate, and interact with their environment, shaping their roles in nature and disease. Microscopy is essential for studying microorganisms since they're too small to see with the naked eye. Light microscopes help us observe shapes and staining patterns, while dark ground microscopes let us see unstained specimens using special illumination. These tools open up the microscopic world, allowing us to explore microbial morphology and behavior in detail. Understanding a microscope's parts is key to using it effectively. Components like the ocular lens, objective lenses, stage, and illuminator work together to magnify and focus light on specimens. Adjustments like coarse and fine focus help us see details clearly. Mastering these parts allows us to observe microorganisms with precision and clarity. Dark ground microscopy is a technique used to observe unstained, transparent specimens, making them appear bright against a dark background. It's particularly useful in microbiology to study live organisms like bacteria and algae, and in other fields like hematology. Other types of microscopes include phase contrast microscope, fluorescent microscope, and electron microscope. The use of microscopes to view tiny objects has a wide array of applications across various fields. These include biological research, medical diagnostics, forensic science, material science, and industrial quality control. Microscopes allow scientists and researchers to observe and analyze structures and materials that are invisible to the naked eye, leading to advancements in diverse areas. A phase contrast microscope is used to observe unstained, transparent biological samples by converting phase shifts in light passing through the sample into visible brightness variations. This technique allows for the study of living cells and their internal structures, such as organelles and cell movements, without the need for staining. Fluorescent microscopes are used to visualize specific structures, molecules, or proteins within cells and tissues, enabling researchers to study their distribution and dynamics. This technique is widely used in biological research and medical diagnostics. Fluorescence microscopy is particularly valuable for live cell imaging, allowing researchers to observe cellular processes in real time. Electron microscopes provide much higher resolution than light microscopes, enabling us to see surface details and internal structures. The scanning electron microscope, SEM, shows detailed surfaces like pollen or blood cells, while the transmission electron microscope, TEM, reveals internal cellular components. These tools are invaluable for advanced microbiological studies. Bacterial staining techniques are crucial for visualizing bacteria under a microscope. Simple staining uses one dye to highlight basic structures, making bacteria easier to see. Special staining targets specific structures with advanced methods, providing more detailed information. These techniques help differentiate bacteria and understand their characteristics. The smallest size human eye can visualize is 0.1 millimeter or 100 micron, and the size of a bacteria ranges between 0.5 to 5 micrometer. They are prokaryotic microbes, have no membrane-bound nucleus and mitochondria. They have a single chromosome as genetic material. They are enclosed in a rigid cell wall made up of peptidoglycan. Some bacteria, along with plasma membrane, contain an additional membrane called a capsule. Bacteria come in various shapes, which helps in their identification. Spherical bacteria, or cocci, can form pairs, clusters, or groups. Rod-shaped bacteria, called bacilli, may form chains or have flagella for movement. Spiral-shaped bacteria include Vibrio and spirochetes. Recognizing these shapes is fundamental for classifying bacteria and understanding their behavior. Bacterial cells have several key components, including a cell wall, cell membrane, cytoplasm, nucleoid containing DNA, and ribosomes. Some bacteria also possess structures like flagella, pili, and a capsule. The bacterial cytoplasm is the gel-like substance enclosed by the bacterial cell membrane, containing various essential components for the cell's functions. It's primarily composed of water, around 80%, and houses structures like ribosomes, the nucleoid containing DNA, and plasmids, as well as enzymes, nutrients, and waste products. Unlike eukaryotic cells, bacterial cytoplasm does not contain membrane-bound organelles. The bacterial nucleoid is the irregularly shaped region within a prokaryotic cell that contains the cell's genetic material, primarily DNA, but lacks a membrane. It's essentially the bacterial equivalent of the nucleus in eukaryotic cells. Plasmids are small extrachromosomal DNA molecules in bacteria. They carry genes, often for antibiotic resistance, and have structures similar to chromosomes. While plasmids don't participate in reproduction, they replicate independently. This ability makes plasmids important players in bacterial adaptation and survival. Bacterial cells have several key components, including a cell wall, cell membrane, cytoplasm, nucleoid containing DNA, and ribosomes. Some bacteria also possess structures like flagella, pili, and a capsule. The bacterial cell wall provides shape and rigidity, which is vital for survival. Some bacteria, like mycoplasma, lack a cell wall and have no definite shape. Gram-positive bacteria have thick peptidoglycan layers, while gram-negative bacteria have thinner layers plus an outer membrane. These differences affect staining and antibiotic susceptibility, making the cell wall a key feature in bacteriology. The bacterial cell wall is primarily composed of peptidoglycan, a rigid layer that maintains cell shape and protects against osmotic pressure. It's a mesh-like structure made of repeating units of N-acetylglucosamine and N-acetylmuramic acid, cross-linked by short peptide chains. The visuals give a closer look at the difference between gram-positive and gram-negative cell wall structure and their difference in composition. Bacterial lipopolysaccharide, LPS, also known as endotoxin, is a major component of the outer membrane of gram-negative bacteria. It's a large amphipathic molecule consisting of lipid A, a core oleosaccharide, and a distal polysaccharide O antigen. LPS plays a crucial role in both the structural integrity of the bacterial outer membrane and the triggering of the host's innate immune response. The bacterial capsule is a protective layer surrounding some bacteria, contributing to their ability to cause disease. Made of polysaccharides or polypeptides, it shields bacteria from the immune system. Techniques like gram staining and India ink help us detect capsules, which is important for diagnosing infections and understanding bacterial virulence. Bacterial flagella are helical, thread-like appendages that enable bacteria to move. They consist of three main parts: the filament, the hook, and the basal body, each with a specific function. The filament is the long, helical structure that provides propulsion. The hook acts as a universal joint connecting the filament to the basal body, and the basal body acts as a rotary motor embedded in the cell membrane. While the overall structure of bacterial flagella, the filament, the hook, and the basal body, is generally conserved between gram-positive and gram-negative bacteria, a key difference lies in the complexity of the basal body, which is directly related to the distinct cell wall architectures of these two bacterial groups. Gram-negative bacteria typically possess 4 rings in their basal body, anchored in different layers of the cell envelope. In contrast, gram-positive bacteria lack the outer membrane found in gram-negative bacteria, and consequently, their flagella have a simpler basal body with only 2 rings. Different bacteria display distinct flagellar arrangements. Monotrichous, meaning a single flagellum at one end, seen in Vibrio cholera. Lophotrichous is a cluster of flagella at one end, as in Spirillum species. Amphitrichous, one or more flagella at each end, allowing directional changes, seen in Alkaligenes faecalis. Peritrichous, flagella cover the entire cell surface, typical of Escherichia coli. Some bacteria are atrichous, meaning they lack flagella and are non-modal, and their presence can be demonstrated with methods like hanging drop method. Bacterial pili are protein structures that allow bacteria to adhere to surfaces, aiding infection. They also play a role in genetic exchange between bacteria, contributing to antibiotic resistance. Notably, pili do not help bacteria move. Understanding pili helps us grasp how bacteria attach and spread, which is key for developing treatments. Bacterial spores are formed during harsh conditions through sporulation. They enter a dormant state with no metabolic activity and are highly resistant to environmental stresses. Typically found in gram-positive bacilli, spores can be identified using special staining methods and serve as important indicators in the lab due to their resilience. Here's a breakdown of the key components of a bacterial spore, from the outside in. Exosporium, This is the outermost layer. It can mediate interactions with the environment and play a role in germination. Spore coat is a thick protective layer of protein, providing resistance to chemicals and enzymes. Cortex, it contributes to dehydration of the core, enhancing heat resistance. Germ cell wall, this layer of peptidoglycan will become the cell wall of the bacterium on germination. Inner membrane, a permeability barrier surrounding the core, similar to the plasma membrane in vegetative cells. Core, the innermost part, containing the bacterium's DNA, ribosomes, and enzymes. The location of an endospore within the vegetative cell is an important characteristic used for classifying and identifying spore-forming bacteria. The position of the spore within the cell, along with its shape and whether it causes the cell to swell, are important characteristics for identifying and classifying spore-forming bacteria. Protoplasts, spheroplasts, and L-form bacteria are all bacterial cell types that have lost or modified their cell walls. Protoplasts are essentially bacterial cells stripped of their cell walls entirely, while spheroplasts retain a portion of their cell wall. L-form bacteria are a broader category of cell wall deficient bacteria that can arise spontaneously or be induced in the lab. Bacterial growth curve depicts the characteristic pattern of population growth of bacteria over time in a closed system. It consists of 4 distinct phases: lag, log, exponential, stationary, and decline, death. These phases reflect the bacteria's adaptation to a new environment, rapid multiplication, stability due to resource limitations, and eventual population decrease. In lag phase, bacteria are introduced to a new environment and are metabolically active but not yet dividing or dividing very slowly. They're adjusting to the new conditions, synthesizing necessary enzymes, and increasing in cell size. Log exponential phase, under optimal conditions, bacteria divide at a constant, rapid rate, leading to exponential population growth. Stationary phase, nutrient depletion and the accumulation of toxic waste products slow down bacterial growth. The rate of cell division equals the rate of cell death, resulting in a stable population size. Decline, death phase. As resources become further depleted and waste products accumulate, the rate of cell death exceeds the rate of cell division, leading to a decrease in the viable bacterial population.